Chapter 3. Mushrooms in their natural habitats. Mushrooms can be placed in 4 basic categories: saprophytic, parasitic, mycorrhizal, and endophytic, depending on how they nourish themselves. However, exceptions abound, since some species employ more than one strategy, making them difficult to categorize. Approximately 8,000 macrofungi, visible to the naked eye, are saprophytic. Around 2,000 to 3,000 are mycorrhizal, and the remaining are either endophytic or parasitic, although more species are constantly being discovered and categorized. The balance of populations can vary drastically with environmental change, however. Deforestation causes a rise in saprophytes and a decline in mycorrhizal mushrooms, for example. Now let's take a short tour through the 4 major categories of mushrooms. Saprophytic mushrooms, the decomposers. Saprophytic mushrooms, the decomposers, steer the course for proliferating biological communities, shaping and forming the first menus in the food web from dead plants, insects, and other animals. Most gourmet and medicinal mushrooms are wood decomposers, the premier recyclers on the planet. Building soils is the primary outcome of the activities of these saprophytic fungi, whose filamentous mycelial networks weave through and between the cell walls of plants. When organic matter falls from the canopy of trees and plants overhead onto the forest floor, the decomposers residing in the soil process this newly available food. Competition is intense. On the forest floor, a single habitat can actually be matrices of fungal networks sharing one space. These fungi secrete enzymes and acids that degrade large molecules of dead plants into simpler molecules, which the fungi can then reassemble into building blocks such as polysaccharides for cell walls. From dead plants, fungi recycle carbon, hydrogen, nitrogen, phosphorus, and minerals into nutrients for living plants, insects, and other organisms sharing that habitat. As decomposers, saprophytic mushrooms can be separated into 3 key groups: primary, secondary, and tertiary, although some mushroom species can cross over from one category to another depending on circumstances. Primary and secondary and tertiary decomposers can all coexist in one location. Primary and secondary decomposers, such as oyster and meadow mushrooms, are the easiest to cultivate. Primary decomposers. These saprophytes are typically the first to grow on a twig, a blade of grass, a chip of wood, a log, a stump, or a dead insect or another animal. Primary decomposers are typically fast-growing, sending out rapidly extending strands of mycelium that quickly attach to and decompose plant tissue. These woodland species include oyster mushrooms, shiitake, and maitake. However, species employ different sets of enzymes to break down plant matter into varying stages of decomposition. Secondary decomposers. Secondary decomposers rely on the activity of primary fungi that initially, although partially, break down plant and animal tissues. Secondary decomposers all work in concert with actinomycetes and other bacteria and fungi, including yeasts, in soil, in the forest floor, or in compost piles. Heat, water, carbon dioxide, ammonia, and other gases are emitted as byproducts of the composting process. Once the microorganisms, especially the actinomycetes, in the compost piles complete their life cycles, the temperature drops, encouraging a new wave of secondary decomposers. Cultivators exploit this sequence to grow the white button mushroom, the most widely cultivated mushroom in the world, Agaricus bisporus. Other secondary saprophytes that compete with compost-grown mushrooms are inky caps, belonging to the family Coprinaceae, which includes the choice edible shaggy mane, Coprinus comatus, and others including the hallucinogenic Peneolus subalteatus and Peneolus cyanescens, and in outdoor woodchip beds, the ambiguous Stropharia ambigua. Industrial growers try to thwart these undesired invaders by heat steaming their composts to temperatures inhospitable to their spores. Secondary decomposers as a group seem more versatile than primary decomposers for dealing with complex assortments of microorganisms, since they have evolved in direct contact with microbial-rich soils. Secondary decomposers typically grow from composted material. The best culinary Stropharia species, the garden giant, or king Stropharia, Stropharia rugoso annulata, is an example of an intermediary between primary and secondary decomposers, since this species first digests fresh debris and then continues to thrive as complex communities of microbes join with it to create soil. Tertiary decomposers. This difficult to categorize group includes fungi found toward the end of the decomposition process. They thrive in habitats created by primary and secondary decomposers over a period of years, often popping up from soils holding little decomposable material. Tertiary decomposers include species of Canosabe, Agrasabe, Mycena, Pluteus, and Agaricus. Tertiary decomposers rely on highly complex microbial environments. The division between secondary and tertiary decomposers is often obscure. Mycologists simply call tertiary decomposers soil dwellers, for lack of a better description. Some mushrooms initially act as parasites, and once they have killed their hosts, they act like saprophytes, growing on their dead remains. Honey mushrooms belonging to the genus Armillaria are good examples of species that grow both parasitically and saprophytically. Parasitic mushrooms, blights of the forest, or agents for habitat restoration. Parasites are predators that endanger the host's health. In the past, foresters saw all parasitic fungi as hostile to the long-term healths of forests. Although they do parasitize trees, they nourish other organisms. Parasitic fungi such as the honey mushroom, which can destroy thousands of acres of forests, are stigmatized as blights. However, more foresters are realizing that a rotting tree in the midst of a canopied forest is, in fact, more supportive of biodiversity than a living tree. Parasitic mushrooms may be nature's way of selecting the strongest plants and repairing damaged habitats. Ultimately, parasitic mushrooms set the stage for the revival of weakened habitats that are too stressed to thrive. Of all the parasitic blight mushrooms that are edible by humans, the assorted honey mushrooms such as Armillaria mellea and Armillaria oysteriae are the best known. One mycelial mat from a honey mushroom, Armillaria bulbosa, made national headlines when a specimen was found in a Michigan forest that covered 37 acres, weighed at least 50 tons, and was estimated to be about 1,500 years old. In Oregon, a far larger honey mushroom, Armillaria oysteriae mycelial mat found on a mountain top, covers more than 2,400 acres and is possibly more than 2,200 years old. Each time this fungus blight sweeps through, nurse logs are created, soil depth increases, and centimeters of soil accumulate to create even richer habitats where once only barren rock stood. For further discussion of Armillaria blight, see page 47. What makes mushroom mycelia different from the mycelia from mold fungi is that some mushroom species can grow into massive membranes, thousands of acres in size, hundreds of tons in mass, and thousands of years old. Many saprophytic fungi can be weakly parasitic, especially if a host tree is dying from other causes, such as environmental stress or parasite infestation. Saprophytes that can take advantage of a dying tree are termed facultative parasites. For example, oyster mushrooms, Pleurotus ostriatus, are classic saprophytes, although they are frequently found on dying cottonwood, oak, poplar, birch, maple, and alder trees. And although reishi, Ganoderma lucidum, is considered a true saprophyte by most mycologists, the Australian Quarantine Inspection Service has classified this medicinal species as a parasite and has banned its importation. Authorities on other islands, including New Zealand and Hawaii, also consider this mushroom a threat to their native trees. Some parasitic fungi behave like saprophytes, such as honey mushrooms, Armillaria mellea, and Armillaria oysteriae, which may be found thriving on the corpse of their tree host. Most parasitic fungi, however, are microfungi, barely visible to the naked eye, but in mass, they inflict cankers and lesions on the shoots and the leaves of trees. Often their prominence in a middle-aged forest is symptomatic of other imbalances in the ecosystem, such as acid rain, groundwater pollution, and insect damage. After a tree dies, parasitic fungi may inhabit the tree, competing with saprophytes for dominance. Since the hosts for some parasites can be short-lived, Natural selection sometimes favors fast growers. Foresters have observed this with Phytophthora ramorum, the cause of sudden oak disease. This downy mildew pathogen can kill an ancient oak tree in days and an ancestral forest in a few weeks, and remain viable on the dead carcasses of its victims, allowing a new staging platform for infection further into the forest. Mycorrhizal mushrooms, fungus and plant partnerships. Mycorrhizal mushrooms, myco means mushroom, rhizal means related to roots, such as matsutake, boletus, and chanterelles, form mutually beneficial relationships with pines and other plants. In fact, most plants, from grasses to Douglas firs, have mycorrhizal partners. The mycelia of fungal species that form exterior sheaths around the roots of partner plants are termed ectomycorrhizal. The mycorrhizal fungi that invade the interior root cells of host plants are labeled endomycorrhizal, although currently the preferred term for these fungi is vesicular arbuscular mycorrhizae, VAM. Both plant and mycorrhizae benefit from this association. Because ectomycorrhizal mycelium grows beyond the plant's root, it brings distant nutrients and moisture to the host plant, extending the absorption zone well beyond the root structure. The mycelium dramatically increases the plant's ingestion of nutrients, nitrogenous compounds, and essential elements, phosphorus, copper, and zinc, as it decomposes surrounding debris. David Perry postulates that the surface area, hence its absorption capability, of mycorrhizal fungi may be 10 to 100 times greater than the surface area of leaves in a forest. As a result, the growth of plant partners is accelerated. Plants with mycorrhizal fungal partners can also resist disease far better than those without. Fungi benefit from the relationship because it gives them access to plant-secreted sugars, mostly hexoses, that the fungi convert to mannitols, arabitols, and erythritol. One of the most exciting discoveries in the field of mycology is that mycorrhizae can transport nutrients to trees of different species. One mushroom species can connect many acres of a forest in a continuous network of cells. In one experiment, researchers compared the flow of nutrients via the mycelium between 3 trees, a Douglas fir, a paper birch, and a western red cedar. The Douglas fir and paper birch shared the same ectomycorrhiza, while the cedar had an endomycorrhiza. The researchers covered the Douglas fir to simulate deep shade, thus lowering the tree's ability to photosynthesize sugars. In response, the mycorrhizae channeled sugars tracked by radioactive carbon from the root zone of the birch to the root zone of the fir. More than 9% of the net carbon compounds transferred to the fir originated from the birch's roots, while the cedar received only a small fraction. The amount of sugar transferred was directly proportional to the amount of shading. An earlier study by Christina Arnbrandt and others showed a similar bidirectional transfer of nitrogen-based nutrients from alder to pine through a shared ectomycorrhizal mycelium. The Simard experiment showed that a common mycelial net could unite 3 species of trees and underscored a remarkable ability of mycorrhizal fungi. Mycorrhizae can keep the diverse species of trees in forests fed, particularly younger trees struggling for sunlight. Now we have a better understanding of how saplings survive in the shadows of elder trees that tower overhead and block out essential light. The fact that a single mycorrhizal mushroom nutritionally supported 2 different trees, one a conifer and the other deciduous, shows that the mycelium guards the forest's overall health, budgeting, and multidirectionally allocating nutrients. Another example of a fungus and plant partnership is the matsutake, which has a unique relationship with the non-chlorophyll-producing candy stick plant. The candy stick gains virtually all its sugars from the matsutake mycelium and the western hemlock and/or Sitka spruce with which it associates. One mycologist I know speculates that the spot fruitings of matsutake on a slope of Oregon's Mount Hood may in fact be from a vast interconnected mycelial colony extending over thousands of acres.
A further example is the big leaf maple, which projects vine-like aerial roots that ascend to the canopy of Pacific Northwest rainforests and are teamed with mycorrhizae. Our understanding of the role of fungi in the forest is far more advanced than the simplistic views held just a few decades ago. Most ecologists now recognize that a forest's vitality is directly related to the presence, abundance, and variety of mycelial associates. A large portion, 1/10 to 1/5 of the total biomass in the topsoil of a healthy Douglas fir forest in the Pacific Northwest, may be made up of mycelium, and even more if we include the endomycorrhizae and ectomycorrhizae that thrive in the canopy. I doubt a forest can be defined without its fungi. Growing mycorrhizal mushrooms has proved to be a greater challenge than first anticipated due to the complex interdependencies in which fungi play a critical role. Once the hurdle of establishing mycorrhizal mycelium has been overcome, decades may pass before a single mushroom forms. Nuances of climate, soil chemistry, and predominant microflora limit our success in cultivating mycorrhizal mushrooms in natural settings. The challenge we face is to tilt the balance so that a species of our choice can take up residence in such a complex natural setting, to design habitats in which it can grow. Species native to a region are more likely than imported species to adapt readily to these designed habitats. Many American growers hope for huge profits when they try to grow European truffles, mycorrhizal mushrooms that sell at very high prices. In an attempt to duplicate the well-established truffle orchards in France, Spain, and Italy, where the renowned Perigord black truffle fetches up to $500 per pound, dozens of growers have tried to cultivate non-native European truffles around the American oaks or filberts on their land. Capitalizing on the desire, several companies now market truffle-inoculated trees for commercial use, and calcareous, high in calcium, soils in Texas, Washington, and Oregon have been suggested as ideal sites for these. One company that seems on top of its game confirms that the tree, inoculated with truffle mycorrhizae, is absent of competitor fungi before shipment. However, I know of only a few successes, one from North Carolina and one from Northern California, that have produced European truffles, and only after more than a decade of effort. In the past 30 years, tissue culture techniques have increasingly replaced the tradition of transplanting truffle-supporting trees. Despite this development, most plantings or inoculations of European truffles beyond their native habitat still fail to produce mushrooms, showing that growing native species is far more successful than growing non-native ones. A truffulateur in Washington recently produced the Oregon white truffle, after patiently waiting for 20 years until the first truffles could be harvested. Nevertheless, commercialization of mycorrhizal gourmet mushrooms has seen little success outside of the European truffle orchard, particularly those of France and Italy. The reality is, though, that our native species of mycorrhizae quickly outcompete the foreign European truffles. Since European truffles like basic, high pH soil, the addition of calcium diminishes competition from native mushrooms, but this alone will not assure success. In New Zealand, where the repertoire of competing mycorrhizae is limited to just a few species, inoculated trees are likely to do better than in regions of North America that are resplendent with hundreds of competing mycorrhizal varieties. One method of inoculating mycorrhizae calls for planting young seedlings next to the root zones of proven truffle trees. The new seedlings acquire mycorrhizae from a neighboring tree, and a second generation of trees carrying the mycorrhizal fungus is produced. After a few years, the new trees are dug up and replanted in new locations. This method has had the longest history of success in European sites where the soils, trees, and fungi are compatible. Another approach, simple and elegant but not guaranteed, is to dip the exposed roots of seedlings into water enriched with the spore mass of a mycorrhizal candidate. First, mushrooms are gathered from the wild, and the spore-bearing surfaces are removed from the fruiting bodies, crushed and immersed in water. Thousands of spores are washed off, resulting in an enriched broth of inoculum. A spore mass slurry from a single mushroom diluted in a 5-gallon bucket of water can inoculate 100 or more of seedlings. Mycorrhized seedlings are healthier and grow faster than non-mycorrhized ones. Even if you are not successful in growing truffle mushrooms, these trees benefit from this pairing with the introduced mycelium. Tossing spores using water as a carrier on the ground above the root zones of likely tree candidates is another method that takes little time and effort. Habitats should be selected on the basis of their parallels in the wild. For instance, chanterelles can be found in oak forests in the Midwest and in the Douglas fir forests in the Northwest. Casting a spore mass of chanterelles into a forest similar to the one where chanterelles naturally proliferate is obviously the best choice. However, the success rate is not high. Even tree roots confirmed to be mycorrhized with gourmet mycelia will not necessarily yield harvestable mushrooms. Fungi and their host trees may have beneficial associations for long periods of time with no edible fruiting bodies appearing. Inoculations of mycorrhiza by one generation of mycologists may not see fruition until the next generation. Chanterelles are one of the most popular collected mushrooms. In the Pacific Northwest, harvesting chanterelles is a controversial multi-million-dollar business. Unfortunately, the gourmet mycorrhizal mushroom species are not readily cultured. Chanterelles demonstrate an unusual interdependence on soil yeasts, making tissue culture difficult. At least 4 organisms must be cultured simultaneously: the host tree, the mushroom, Pseudomonas bacteria, and soil yeasts. Not only do other microorganisms play essential roles, but the timing of their introduction is also critical to success in the fungal theater. Many experts believe that decades will pass before the plantations growing mycorrhizal species like chanterelles mature to a productive state. No one has yet grown chanterelles to the fruiting body stage under sterile laboratory conditions, although greenhouse-grown pines have produced chanterelles after inoculation. In 1997, Eric Donnell, accompanied by F. Camacho, was the first to successfully cultivate a chanterelle, fruiting mushrooms with a potted 16-year-old pine seedling in a greenhouse. Soon thereafter, Donnell patented this particularly vigorous strain, which showed commercial potential. Field tests in 24 locations revealed chanterelle mycelium in the seedling's root zones 2 years after inoculation. Unfortunately, he cannot stop grazing animals such as deer, squirrels, and beetles from foraging and disturbing his crops. More recently, Donnell started a Swedish company called Cantharellus AB to commercialize this breakthrough mycotechnology in the creation of chanterelle orchards. His group has planted thousands of trees with the chanterelle mycelium in an attempt to create mushroom plantations that produce mushrooms within a decade of planting. For the time being, only the patient may want to invest in mycorrhizal plantations. Given the long time involved in honing laboratory techniques, I favor the low-tech approach of traditional methods of planting seedlings adjacent to known producers of chanterelles, matsutake, truffles, and boletus, and then replanting the seedlings several years later. In this way, we can value the forest not for its quantity of harvestable lumber, but for its potential to harbor mushroom colonies. Mutualistic species, fungal partnerships. Mutualism occurs when 2 or more organisms work directly together for their mutual benefit, usually to prevent infestation by parasites and gather nutrients. Many organisms, from plants to ants, seek fungi as protectors and vice versa. This rapidly expanding field of study is one of the most exciting in mycology. Insects and mushrooms. Many insects use mushrooms as platforms for incubating and feeding their larvae. One of the first cases of mutualism to be noticed was the interrelationship between mushrooms and termites. The mushroom genus Termitomyces includes several species of mushrooms associated with terrestrial termite colonies, especially in the tropics. Termites construct their nests with organic matter and cultivate mycelium. When abandoned, these mycelium-rich nests sprout delicious mushrooms. Since certain fungi function as natural bactericides and fungicides, Some insects engage them as allies in an effort to counter infections from hostile bacteria and other fungi. Studies at Oregon State University show that attine ants, which include leafcutters, grow Mycelium as a host for a benign bacterium that produces an antibiotic against destructive microfungal parasites, and they also feed Mycelium to their larvae. This complex partnering has allowed lineages of attine ants to survive for more than 50 million years and to establish massive colonies, numbering in the many millions of inhabitants. The mutualism between ants, mushrooms, and bacteria is a useful model for how we humans can live in closer harmony with our environment. Both ants and people benefit from the guardianship of mycelium. By partnering with fungi, many organisms, including humans, can resist disease. Snails as fungus farmers. Snails and slugs love mushrooms, an unfortunate situation for many of us mushroom lovers. Some snails enlist fungi to help them digest plants. Silliman and Newell found that a seaside snail, the marsh periwinkle, damages and then defecates in certain grass, where a particular fungus soon grows. Days later, the snails return to the grass, now overgrowing with fungus, and consume both fungus and plant. Grasses without the snail-enabled fungus grew 50% faster, but were less appealing to feeding snails, whereas the plants covered with fungus were more palatable and nutritious for the snails. As you can see, the snail and fungus relationship affects other species in the marsh environment, such as grasses. Endophytes, mutualistic symbionts. Endophytes are primarily benevolent non-mycorrhizal fungi that partner with many plants from grasses to trees. Their mycelia thread between cell walls, but don't enter them, enhancing a plant's growth and ability to absorb nutrients while staving off parasites, infections, and predation from insects, other fungi, and herbivores. Generally, endophytes are not true saprophytes or parasites, but are a class of their own. In contrast to mycorrhizal fungi, many endophytes grow well under laboratory conditions, so we can make spawn by using these methods like those used for saprophytic mushrooms. The vast majority of endophytes are undescribed, and some appear to have lost the ability to produce spores, living vegetatively in a continuous mycelial state. Most endophytes described thus far are ascomycetes. One example is Pesicula aurantiaca, a small cup-like mushroom that lives on healthy alder trees. Like many endophytes, this fungus is dimorphic, expressing itself in two forms, with one being an asexual mold. Endophytic fungi are especially skilled at producing specialized mycotoxins, often alkaloids, a class of compounds that includes toxic cyclopeptides and serotonin-like tryptamines, such as psilocybin. Endophytes hosted by grasses are similar to the ergot fungi, whose alkaloids prevent their hosts from insect attack. Endophytes in large crabgrass, for example, appear to produce toxins that kill fire ants, and those in grasses such as the darnel weed cause sleepiness in cattle and horses, a fact long known to ranchers in Central America. However, Stanley Faith suggests that varying levels of alkaloids in plants may not yet afford consistent protection against herbivores. Because some grasses produce more mycotoxins than others in the same habitat, cattle may sometimes get a chemical cocktail, but other times not, making it more difficult for them to learn which grasses to avoid. Nevertheless, endophytes, which were once thought to be pathogens, are increasingly viewed as engaging the plant in a mutually beneficial relationship. In a 2003 experiment in Panama, researchers found that when endophyte-free leaves from the chocolate-producing cocoa tree were inoculated with endophytes, leaf necrosis and mortality declined threefold, suggesting a biodefensive effect is possible against other pathogens, such as Phytophthora, the genus responsible for sudden oak death, a disease devastating California's native oak population. Spores from endophytes compete with many other free-flying fungal spores. According to one estimate, more than 10,000 spores of fungi land on each leaf per day. Amidst such competition, friendly fungi taking up residence is actually an asset to plants otherwise subject to pathogenic assault. Increasingly, mycologists believe that endophytic fungi may have co-evolved with hospitable plants. Wheat farmers benefit from the endophyte Piriformospora indica. The basidiomycete of this species has yet to be identified, so it's referred to as imperfect. In the mycological world, this means that the fungus has no sexual phase or the sexual phase has not yet been discovered. This species is a root-based endophyte that promotes the growth of wheat shoots and roots and is capable of increasing leaf and seed production by more than 30% while shielding roots from infection by pathogenic microbes. Furthermore, seedlings paired with this mutualist successfully germinated 95% of the time compared to only 50% of the time for seedlings without this species. Root and shoot mass also doubled. This species has also demonstrated growth-enhancing properties when paired with maize, tobacco, and parsley. This fungus is easy to cultivate in the laboratory and widely coexists with many grasses. Clearly, pairing this and other endophytes with agricultural crops can increase yield, decrease disease, and reduce the need for fertilizers and insecticides. Endophytic fungi may have other practical applications in agriculture. Joan Henson and other researchers filed a patent application using a Curvularia species isolated from grasses in the geothermal zones of Yellowstone and Lassen Volcanic National Parks. This fungus qualifies as an extremophile, a thermally tolerant species that grows at the far fringe of temperatures where life can be found, and confers some tolerance to drought and heat to the host plant. Henson's research showed that grasses inoculated with this endophyte survived temporary exposure to extraordinarily high temperatures, 158 degrees Fahrenheit, or 70 degrees Celsius, while those without shriveled and died. Although wheat did not survive in their experiment, it did demonstrate increased drought resistance. When watermelon seedlings and mustard seedlings were dusted with Curvularia spores, the spores germinated and inhabited the young plants. After the endophytic fungi became established on their hosts, researchers exposed the seedlings to extremely high temperatures, 122 degrees Fahrenheit or 50 degrees Celsius. The seedlings with the endophytic spores survived prolonged exposures, but the same types of seedlings without endophytic spores died. The discovery of the Curvularia spores' effects on plants may explain the biological toolset for mycorestoration, possibly even drastically expanding oasis environments and countering desertification. Some woodconchs, once seen as parasites on trees, may in fact be symbiotic endophytes. Baum and others report that the Basidiomycetes polypore, Fomis fomentarius, the tinder polypore or Iceman mushroom, can operate as a non-saprophytic endophyte in beech trees. This well-known polypore is commonly found on beech, birch, poplar, alder, maple, cherry, and hickory, trees that thrive in the boreal regions of the world. In the Baum experiment, healthy beech wood was cut into sections and incubated in culture dishes for 8 weeks, whereupon fungal cultures of this conch emerged. A number of isolates of Fomis fomentarius were identified from the wood sections, and some proved to be genetically different strains. The fact that one species can perform separate but complementary functions in the forest suggests that the species may play a larger role in the forest than is presently understood. How many other perennial woodconchs do the same? I once received a call from a manager of a chestnut orchard in Quebec who told me that trees sporting chaga, the aerial sclerotium of Inonotus obliquus, were resistant to chestnut blight. Sclerotium is a compact mass of hardened mycelium with stored food that can sometimes become a detached entity. When he made a poultice of ground chaga and packed it into the lesions of infected chestnut trees, the wounds healed over and the trees recovered free of the blight. This leads me to wonder whether Inonotus obliquus can operate as an endophyte, as does Fomis fomentarius, and whether these species or others like them could defend host trees against invading parasites. Many other mushrooms may be endophytes, including guild species. For decades, mycologists have been mystified how Psilocybe cyanescens, a psilocybin mushroom, can suddenly appear when trees are chipped into landscape mulch. A mycologist friend of mine had a truckload of fresh, mostly alder chips delivered to his house in the spring. Soon thereafter, his mostly conservative friends took some home for mulching, and that fall, most of their mulch beds were fruiting with hundreds of potent psilocybes. Where could they have come from? My mycologist friend wondered. The only plausible explanation is that the mycelia were already in the wood, above ground, while the trees were alive. Perhaps Psilocybe mycelium can be endophytic, but delays fruiting with mushrooms until the trees make ground contact. When engaging fungal allies, the mycorrhizal, saprophytic, and endophytic mushrooms, plants benefit in 3 ways. These complementary mycological systems help plants survive starvation, dehydration, and parasitization. The richer the fungus-plant partnerships, the more organisms the habitat can support. Masanobu Fukuoka, the farmer scientist, ecological visionary, and author of The One-Straw Revolution, understood that scientific reductionism failed to reflect the biological synergisms, processes that are still far beyond most of the most sagacious scientists today. He invented seed balls, clay soil pellets rich in microbes and weeds, designed to jumpstart weakened habitats. Although Fukuoka did not select the populations of fungi in his seed balls, he appreciated their contribution. One of our tasks is to better Fukuoka's pioneering method and customize it for myco-restoration projects. Beginning with one straw, the saprophytes lead the charge, followed by mycorrhizae and endophytes. As caretakers for future generations, mushroom communities surrounding trees govern habitat progression. I believe fungi have evolved to support habitats over the long term, protecting generations of hundreds of years in the future. Saprophytic mushrooms gobble up debris fallen from the trees and prevent invasion by parasites. The mycorrhizae channel nutrients, expand root zones, and guard against parasites. Similarly, endophytic fungi, less well understood, chemically repel bacteria, insects, and other fungi. After hundreds of millions of years of evolution, fungal alliances have become part of nature's body politic. It is time for our species to partake in this ancient mycological wisdom.